But I thought what I would do is just give a little bit of background about the whole scientific process that's around publishing scientific works. So I'll kind of set everything up, give you some background there, and then we'll dive through a publication, not the latest one that Dr. Nettles teased, but yes, we will have a human clinical study coming out literally any day now centered around Protandim NAD Synergizer. But I thought I'd go back and revisit the first publication that came out as well. So that's kind of the flow for today. A lot of this is just going to be on the fly. I hope RiFi was able to send out the publication to anybody who wanted it. But with that, we'll kind of jump right in, and I'll kind of maybe set the stage of what's happening even before we get to the point of having to publish some scientific works. So let's say I'm working in a laboratory. And it's a interesting little, I don't know if it's a dirty secret of academia, but most laboratories, they're run by a professor. That laboratory will be full of graduate students, research assistants, associates, postdocs as well. So after someone gets their PhD, they'll stay on to continue to do research until they find a big discovery where they can start applying for jobs as a professor at an institution. But these laboratories, they tend to focus on one thing. So a laboratory becomes really specialized, really in one small niche area of science, and especially my field of molecular biology. And a lot of that has to do with grantsmanships and the way the scientific process works, and science kind of building on science. So these professors really kind of start to pigeonhole themselves because they need to get grants, and the only way to get grants is to further the research that they already had a grant on. And it's so interesting when I hear that research that comes out of industry may not be as acceptable or as honest or truthful as research coming out of an academic institution. But I do take exception to that. I mean, maybe what we call profitability, they call grantsmanship. So they're always doing research with a bias towards their next grant. And of course, we want to make money in a corporation. So I'm working in a laboratory under the context of whatever field the professor wants to study in his laboratories. And then at some point, whether it's reading the scientific literature or if it's conducting my own experiments, I'll come up with this experimental question. Gosh, I wonder what happens if we did X, Y, or Z. And we set up the experiment and we do the studies, get the data back, analyze it. And then if we're able to find something new, or even if we're able to repeat somebody's work who's already been done in the scientific literature, that's really worthy of publication. So once we think we have either a new discovery or we're somehow adding to the bigger picture of the scientific literature, say, okay, we're going to publish. So what do we do at that point? So the first thing we want to do is decide how new or novel is the research that we were conducting, and especially the data and the conclusions we're able to draw from those research studies. And then so based on how new and exciting it is, we then determine which scientific journal we want to submit that work to. I bet most people on this call have never heard of something called an impact factor. So I pulled up a little screenshot from the web of kind of explaining what an impact factor is of a scientific journal. So the impact factor is really the prestige level of a scientific journal. And the way an impact factor is determined is taking the total number of citations that have been cited from an article over the past 2 years divided by the total number of potential articles. So the reason this becomes important is because those journals that have the highest impact factor or prestige are the ones that tend to publish the newest and most exciting research in a field, and research in a field that's really going to change kind of the face of science or the course of a research agenda. So down here lower on the page, you may have heard of some of these journals, but these would be the most prestigious journals that are out there today. So there's the journal Nature. I bet most of us have heard of the New England Journal of Medicine. There's the journal Science. The Lancet is another medical journal that rivals the New England Journal of Medicine. There's the journal Cell. And when you're in those top five are really constantly trading spots. So really, to be able to publish in one of these top 5 journals is an incredible accomplishment, and it's really good for a researcher's future career prospects. And we get into some other ones you've probably heard of, but the Proceedings of the National Academy of Sciences, or PNAS, again, a really prestigious journal that's ran by scientists that have been inducted into the National Academy of Sciences, Journal of Clinical Oncology, and then Chemical Reviews. So you can go and pull up an impact factor for any journal. And as a scientist, we do always want to try to publish in the highest impact factor journal that we can. That just brings instant credibility. It's selfishly, like I said, it's good for our careers. But besides impact factors, some of the other things that we need to consider are, well, let's publish, in a journal that's at least relatively related to the field of study of my research. And then we'll also look whether or not a journal might be publicly available or whether it's behind a paywall. In an academic setting, that doesn't become so important because most academic settings give all their students and faculty free access to any journal that's out there. But we know that's not the case. And some of these journal articles can be incredibly expensive for someone who may not be in an academic setting. So in our case, quite often what we'll do is we'll aim for a journal that's publicly accessible so that you all can easily have access to the journal. Now, once we've decided what journal we're going to publish in, we then send a rough draft, and it's interesting. We just put them together in a Word format, and I just happen to have, this isn't related really to anything, but here's, I don't know if you can really see it, but it just says accepted manuscript, but you can see it was all put together in Word. So we'll send a Word document off to the editor, and the editor, based on the research that's being conducted, will then send it out to a small handful of experts in the field. And so then those experts in the field, and it can be anywhere from 2 to 5 to 7 different reviewers, they'll call them, and they go through it with a fine-tooth comb, and they're literally trying to come up with any excuse they can to reject the publication. So, I mean, they're going through it hot and heavy. And again, this is an important thing to consider when you're considering corporate research versus academic research. It doesn't matter which institution that you're from, which type of institution you're from, that same scrutiny is in place no matter what. So I would even argue that scientists that are in a corporate setting probably even have it harder than somebody in an academic setting, because the reviewers will instantly think, oh, there's a bias coming from this organization because they want to promote a product. So I feel like they really put the screws to us when we're trying to submit research. So that work submitted to the editor, he quarterbacks it out to experts in the field, and then those experts evaluate the quality of the experiments. Were the right controls put in place? What does the data look like? What statistics were ran? Statistics is incredibly important for the data analysis, and it is relatively easy to manipulate numbers. I think we've all heard and experienced that. So actually, your statistical methods are incredibly important. Did you draw the correct conclusions? And then how does all of this research fit into the larger context of the scientific literature, among many, many other things? So they're going through it with a fine-tooth comb. And then at that point, they will give a recommendation back to the editor that this publication will either be accepted with revisions. So maybe there was an additional control or something I needed to throw into my experiment. Maybe some of my experimental methodologies weren't so buttoned up. Maybe I got a little aggressive in the conclusions that I was making, overextending the research. So for a number of reasons, they can say it's been accepted with revisions. Sometimes they'll even make you go back and add different experiments into the study to try to get a more rounded approach to whatever experimental question you were trying to answer, or they can just outright reject it. And then we start shopping them again to different journals. So we kind of start with our dream impact factor journal and kind of start working down from there. So I don't know if that kind of makes sense and then sets the stage. If your publication is accepted, you usually get kind of a publication date. It'll be available online first, and then it will also appear in an actual hard-covered, well, soft-covered bound journal in a library somewhere. So Dondi, how do you feel? Does that kind of set the stage for going through this first publication?
Yeah, absolutely. Just a question, when you're talking about when they send it out to reviewers and it comes back either accepted or accepted with revisions or rejected, now you've got to go shop it out to another journal. What's the statistic, if there is in your experience, what's the statistical probability that on first submission that it comes back either accepted or accepted with revisions versus rejected?
It's very rare they come back outright accepted. It's very, very rare. I mean, I would put that probably in the 1% category. Accepted with revisions, again, depending on how high of an impact factor journal you're shooting for, you can kind of determine your own odds there. So I do know, I don't know the specific number, but I do know that for journals like Cell, Science, Nature, those rejection rates are incredibly high. We may have better luck getting into the MBA, Dondi, than actually having a paper submitted to one of the, or accepted by one of those journals. So those rejection rates are incredibly high.
And with regard to, say, getting into a Nature or a Science, is it something that the more likely that this is a groundbreaking discovery or something relatively new, does that increases the likelihood of acceptance?
Absolutely. So when you make the biggest discoveries in the lab and you just think, oh my gosh, this is going to change the course of future research, those are exactly the kind of studies that those publications want. And it's an interesting feed forward cycle because they're already prestigious. So they basically get access to the best research, the most cutting edge research. And they also are in a place where they can be choosy. Because if they can determine to the best of their abilities what research is going to change the course of a research agenda or a topic, of course they want those studies because they know that that study is going to be referenced over and over and over again in that field. So they've got an agenda. The researchers submitting to those journals have an agenda, and it works, and why those 5-ish or so journals are just consistently at the top. Kind of like applying to Harvard or MIT or Carnegie Mellon, someplace like that.
Got another question on acceptance versus rejection rates. If something is applicable on a broad spectrum, say this, like NRF2 activation as an example, it affects every human being who's breathing air, versus something like the disease progeria, which is the disease where children age extremely rapidly, and most of them look like they're 90 years old when they're 17, which is a very, very obscure, very rare kind of a disease. So is there likelihood to accept something based on broad spectrum application versus something extremely rare?
Yeah, it'll again depend on the impact that journal thinks that research is going to have to the field. So with NRF2 being discovered back in 1994, there's been quite a bit of work that's done on NRF2 right now. So my guess would be, unless something just really profound is discovered around NRF2, the likelihood that you'll see some NRF2 publications in there, in those journals, is probably pretty low. But something like progeria, where you're using kind of as a surrogate for, in this case, a disease that doesn't affect very many individuals. But if they were able to find, say, the specific genetic link to progeria, that might be worthy of publication in that type of journal. We'd say similar things about autism or insert your disease there. So it just really depends on the impact that research is going to have on that field of study moving forward.
Gotcha. And just one comment, when you're talking about acceptance rates being around 1%. For a company like LifeVantage to be sitting in a place where we have 31 peer-reviewed studies, 28 of them on PubMed, that would you say that's kind of that's an anomaly in number one, our space in network marketing, but pretty much across the board in supplements and nutrition?
Absolutely. I've never seen anything like it in the industry, to be very candid. I mean, nothing that even comes close to, like, those studies around Protandim NRF2 Synergizer. I mean, we're just light years ahead, you know, with the 28 or so we can find in PubMed and the 31 studies that we have done totally. Actually, you brought up an interesting point too. Just it sparked another thought. But then, like, what about PubMed? We tell people to go to PubMed all the time, but what is PubMed? And PubMed was originally founded by our United States National Institutes of Health, and the whole goal with that website of PubMed is to collate all the medical and scientific research that's done absolutely in the United States, but even around the world. So it's quite common you'll find articles that are written in another language. So they do the best that they can to collate all the biomedical research that's out there. And then if you get governmental grants, I believe now without exception, they require that you are going to publish work in a journal that will for sure be collated into PubMed as well. So PubMed is this amazing resource. Gosh, I looked recently, but it's millions of articles are collated in PubMed. And so that's where it's a really easy place to go find scientific works around any topic that's related to human health, wellness, or disease.
Yeah, and one point I wanted to make, and why I asked you the question as far as how we rate ourselves as having 28 studies on PubMed and how we rate ourselves to our competitors in the industry and just in the dietary supplement market as a whole. No one even comes close, you said, but to the people that are critical of our science, and so people that are who are scientists or researchers or doctors, they'll look at it and go, you only have 28 studies. When you have 1,000, come back and let's have this discussion again. They don't really, because they're comparing their reality to our reality, which is like comparing apples to oranges. So I would just tell you guys, if you've experienced that, don't lose faith and don't get disheartened because someone who's a professed expert tells you only have 28 studies. That's 28 more than everyone else. So just wanted to make that point.
That's a wonderful point, Don D. And what's so interesting is that's many times and probably many fold times larger than a pharmaceutical company would need to be able to bring a drug to market, for example. Yeah. So to put that into context, you're lucky to see a couple, if not a small handful of publications around a pharmaceutical drug. Yeah, so you're right. The level of scrutiny is is high in the nutritional space. I think LifeVantage has done an amazing job of rising above it all. And I think another interesting point is those 28, 31, however you want to count, studies have been done on the final formulas of Protandim. So if you start throwing into the mix research done on green tea, turmeric, milk thistle, right, et cetera, that breadth of scientific literature just gets astronomically large. But we're talking on the final formulas, which is bonkers.
Yeah, it definitely sets us apart.
Yeah, absolutely. Well, if you're okay, why don't we jump right into the article that we wanted to discuss today? And like I said, maybe a little bit of grace on my part as we try to navigate this. I thought I had all my screens ready. I apologize.
You guys are all witnessing what it looks like when 180 million brain cells are all functioning at the same time.
What does my face look like? I should pay better attention. All right, here's the article we want to talk about today, and it's centered around Protandim NAD Synergizer and the core blend that's there. So a couple of things just to orientate you all before we jump in, and maybe I'll read the title just to get started. But a teacrine-based supplement increases cellular NAD levels and affects biomarkers related to sirtuin activity in C2, C12 muscle cells in vitro. So quite a mouthful, but quite often what they will do in the titles of these publications is really put the main conclusions, and then they'll sprinkle in some other key words to help make them easier to find. But I kind of want to point out, besides the title, some of the other key sections that are in literally every scientific paper. So the first would be the abstract. Okay, so abstract. The abstract is really kind of like the outline for the rest of the study. So it's going to have a short, introduction. In this case, it's 1 sentence long. They're going to talk about the experimental design. They're going to talk about the methods, the results, the conclusion, and the discussion, all kind of right here. Then another key point is, or a key part of the publication is the introduction. And so this is a really good way to give yourself some background on the topic that's going to be discussed. And they try to put this research into the context of what is already known in the scientific literature. Then if we go down to the methods, to be quite candid, I generally don't spend a lot of time in the methods. For this paper, there was a couple times I had to refer back to it just to get a feel for what they're doing. But generally saying, researchers are using well-established methodologies to be able to conduct their research. So there's just a lot of established techniques that we know exist to measure whatever it is that we want to measure. So I don't find this necessarily to be helpful, but it's there in case you ever just want some more information and to figure out exactly what they did. And the whole point of that method section is that another researcher theoretically should be able to pick up that section of a scientific journal and be able to repeat those studies. And then we get into really the key part of a scientific manuscript, and that's the results. And that's where they lay out what exactly was found. And so you'll see there'll be graphs, tables, charts, pictures, whatever it takes to make the case that their experimental question was valid and that they found something new. And then the discussion is really where these researchers are putting their research into the context of the larger scientific literature. So what's already known, what did they find, and then what are also necessary next steps. And then we have the conclusion. They just brass tacks tell you what they found. Pretty straightforward, quite often pretty short. And then the reference section. So any good scientific manuscript will have an extensively referenced introduction and discussion, especially methods as well can be here. But if you want more information or you want to keep the authors honest, this is a good place to go ahead and do that. Now, I'll just maybe give you the punchline up front. So if I need to race through an article for whatever reason, the way I'll kind of cheat my way through it and get through a scientific publication as quick as possible is I will read the abstract, because that basically gives you the conclusion of the study. I'll read through the introduction just to get that background and context, and then I'll quickly scan the figures and then into the discussion. And that really gives you a very solid picture about what this article contains. And I think we can cheat on that front sometimes because we can rely to a large extent on that peer-reviewed process. But here's how I'll go and tackle a publication. So first is the title. And the title actually tells me a lot. It tells me that diacrine must be a prominent part in the supplement. It's telling me they're measuring NAD levels. In fact, it's telling me that this supplement, whatever it is, increased cellular NAD levels and also affected other biomarkers related to sirtuin activity. And then it also tells me what experimental model they were using. And in this case, I happen to know what C2, C12 cells are. So they're muscle cells from mice that are immortalized. And as crazy as it sounds, you can just buy them from different organizations. They're immortalized, so they're really pretty durable cells that grow easily in a laboratory. but have muscle characteristics. So it is really very critical that researchers are choosing the best experimental models to answer the questions that they want. And the reason they chose muscle cells, which in my opinion is a good choice, is because muscle tends to have the highest levels of NAD. So you want to go to the source, whereas other tissues, maybe if we use cells from liver, maybe they wouldn't have as high of an NAD level. So we want to make sure we're using the right experimental methods to answer the questions. And then I'll just come down. Well, I should talk about, I will also look at the authors just to see if there's anybody I know. I want to see what laboratory it came in. And then something we don't talk about quite a lot, but there are two important authors on this list. And the first is going to be the first author. We call him or her the primary author. This is usually the graduate student that's in the lab actually doing the work. These are probably people that maybe they had expertise in a particular method, so they jumped in to help collaborate, but in some way they provided a unique function, scientific function to this publication. It really is bad form just to throw your buddies on there. There's some pretty strict criteria of who can be an author on a manuscript. And then the other important author is the last author, and that's who we refer to as the senior author. And this is usually the principal investigator or the professor that's running the laboratory. So they both get the most credit for it. So you'll always want to have your name first or last whenever possible. And believe me, it's always best to set up this author order ahead of time, because I have seen some drag out, knockdown fights to try to figure out who's first author, who's senior author, and what order comes in between. But if everyone agrees on it up front, you save a lot of heartache on the back end for sure. So then I'll also look, obviously, to look at the dates. So when was this published? But the year doesn't usually bother me. I mean, science doesn't have an expiration date. It's not a bottle of milk. So if a study was done well 100 years ago, it still has validity today. Of course, some of the techniques have advanced and we can draw better, maybe better, stronger conclusions. But really the date should maybe just be kind of a secondary thought. We talked about the abstract briefly. It's really the outline for the study. So you can get a pretty good feel for the conclusions that they're able to draw in the study. The introduction, like we talked about, is fully referenced. So as you're going through here and maybe there's a topic you're not that familiar with, or you want a little bit more background on, you know, with these paperless PDFs, these are all hot linked now. So it's really easy to jump around and get some quick background on any given topic. And then what ends up happening, and a good little cheat again, is usually the first couple words or sentence will tell you what that paragraph's going to be about. So in this case, it's skeletal muscle aging. Here they're going to put some background around sirtuins. Here they're going to talk about the nutrient teacrine. And then they always end with what we call a gap in knowledge. So that gap in knowledge is going to be right here. Okay, so it is currently unclear how teacrine-based supplements affect the biomarkers related to these processes in skeletal muscle. So that's really the setup for the experimental question that they're asking here. Therefore, the purpose of this report was to assess how teacrine-based supplements affect markers of sirtuin activity and mitochondrial biogenesis in skeletal muscle. So that's the questions they're trying to answer. So then, like I said, for the most part, unless I need, I'll skip over the method section. I might briefly read the headers or the titles to see if there might be a new method I'm not familiar with. But other than that, I do spend a little bit of time here on the statistics. And, you know, Dr. Nettles and everyone on the call, when we see this P equals less than 0.05, does everybody know what that really is referencing? And it'll become an important point here in a minute. I see some heads shaking no. But what a p-value is it's really, it's statistics or odds. And so you want some level of confidence that the results you're finding aren't just happening because it's a random nature. So for whatever reason, some say it's pretty arbitrary, some have good mathematical reasons for it, but generally in biological sciences, we want a p-value of less than 0.05. But what that is really telling us is that if we repeated an experiment 100 times, 95% of the time, we would get the same results. And we have a 5% likelihood that these results are random. So that gives us pretty good confidence that when we see a p-value of less than 0.05, it's not a random result, and that you can put, at least some high level of faith into the results. And then of course, the smaller that number gets, the more confidence we can have. So here, really, like I said, kind of the way you can cheat your way through these papers a little more quickly is just go to the figure and then read the figure legends. So that's basically telling you really quick a method and then what they were measuring. But I can tell here in figure 1, they're measuring mRNA levels. So just like we talk about flipping switches, they were measuring the same thing. So does this teacrine-based supplements flip the switches on, in this case, the sirtuin genes that they were looking at? And you can see at both 3 hours and at 24 hours, there was an increase in those switches being activated on the sirtuin genes. And publications don't usually do it this way, but they put the control second. Usually the standard is control first, and then it's easy to see the change. So you really have to orientate yourself with the figure legend. So make sure you're looking at the right thing, and then also pay attention to the different axes. So the x-axis here is what they're measuring. And then this is how they're quantifying it. So measuring the actual how active a gene is in its expression. And then they went on to measure how much or how active those enzymes or proteins were. And here, in my opinion, they may have missed this step. They didn't actually measure the amount of protein there. They just wanted to look at how active was it there. And for sirtuins, I think we can give a little bit of leeway here to the authors, because really it comes down to brass tacks, and sirtuins are either on or off, and it doesn't really matter how much of that protein is there, but they're able to see that over the course of 24 hours and as soon as 3 hours after adding these nutrients to the cells, they also see the activity of these sirtuin proteins increase. I'll come down to the next figure, figure 2, same thing. So looking at gene activation again through mRNA, and then looking at something called CS activity. Well, gosh, sometimes acronyms can get us in trouble. So always make sure that they're referring to what I think they are. So there's almost always a legend as well in the figure caption that will tell you what CS activity is. And I just happen to know it's a marker for the number of mitochondria that are in cells. So what they're able to look at is, again, some different gene targets, looking at the x-axis, looking at the gene activity. And the other thing I failed to mention in that first figure is you also want to be looking for these little marks. So they can be asterisks, pound signs, they can be a lot of different things. But if those are there, that means that that result is statistically significant. Okay, so that p-value is going to be less than 0.05. So you see here, even though there's a change, it's not statistically significant. So we don't have the confidence that that's a real result. If that makes sense. And again, just maybe for your reference, what are these funny little whiskers that are standing on top of the bar graphs? And again, we can go back up to the methods to find out what they mean. And here it tells us in the methods that they're measuring the standard deviation. So they're looking at the population. They're only looking at one standard deviation. So we know that this bar covers the response of about 68% of the data that was received. Then you can go to 2 standard deviations and get 95%, and then 3 deviations gets you 99.7%. So it's a good way to get the spread of the data, or how variable was the data in an experiment. So tighter error bars are generally considered to be better types of results, where this could have been noise or something could have gone wrong in your experiment. So here was one where I had to go back to the methods to figure out what these were. I didn't know what these two orange, pink, fuchsia boxes were. It doesn't show up very well on the actual printed copy, a little bit better on the digital version. But what it is, it's just pictures of the cells growing in a plate, and they wanted to get the point across that the supplement doesn't make the cells look any different. And that's important, because if a cell in culture, they are really, really finicky. And if a cell in culture doesn't like something, they can kind of shrivel up or ball up. And so here they're saying, no, but the control and the treatments, they look very, very, very similar. They did a fun little experiment where they looked at total protein levels. So protein, to actually make protein within a cell, takes a tremendous amount of energy to make. And so it's generally considered that the more protein there are in a group of cells, the healthier the cells are. So in this case, they saw more protein in the teocrine treated cells versus the control cells, implying that these are healthier cells. And then here in figure 3b, they looked at actual protein level concentrations. And these are pretty fun little experiments to do. You see these everywhere in molecular biology and cellular biology. These are called Western blot analysis. And I found a kind of a cool little schematic. That kind of quickly explains what this is. And it's cool how you're using science to do science. So it turns out that proteins have positive charges and they have negative charges on them. So what you can do is kind of grind up your cells so you just get everything into basically a homogenized liquid. Then what you'll do is mix it with it's usually a blue glycerin base. So you'll take a little bit of your extract that's been all ground up, a little bit of glycerin just so it makes it heavy. And then what you'll do is put that, try not to use scientific words, but you'll put that mixture into the top of a gel that's sitting inside of some water. So at the very top here, you're filling the wells with your extracts from these cells. So this is all in water, a little bit of water down at the bottom. And these gels are really like gelatin or jello. And you can buy different thicknesses, or I should say different densities based on the percentage of the stuff that's making up the gel. So in this case, you can see here, it's a polyacrylamide gel, so a plastic type of gel. Then what you do is hook some electricity up. You put the negative on top, the positive at the bottom, and just like a magnet can repulse another magnet, when that current is applied, those proteins start to run away from the negative charge and towards the positive charge. But because they're in this gel matrix, they have to kind of scramble their way through this matrix. And so the smaller the protein, the more quickly it can navigate that matrix, and the bigger, heavier proteins stay up at the top. And then just to make sure you're running your gels for the right period of time, what you can also do is use what they call a molecular weight marker to put on one side of the gel. And you can tell that maybe when the purple band gets down towards the bottom of your gel, that's time to shut off the apparatus and conduct the rest of the experiment. Then we use some antibodies that bind to a specific protein, and we can confirm ultimately that these are, let's say we're looking for this SIRT1, we know it's 120 Dalton sized protein. So it's in the right spot on our gel, and then we can use the antibodies to actually help visualize the protein. So the antibodies, just like they work in our own bodies, go and bind to the protein, and then they're tagged with some sort of either light, fluorescence, or some other chemical reaction that we can actually visualize. And so here they are looking at some of these different protein levels to be able to determine that, yes, indeed, these were increased not just in gene expression, but also in protein concentration as well. And then usually the important figures get down towards the bottom. So these are kind of setting up, but really, I think brass tacks, what we want to know is, can this supplement actually increase NAD levels? And so when they did look at the NAD levels, they saw an increase in NAD plus. They also saw a slight decrease in a related molecule called NADH. And then also the ratio was increased of NAD to NADH. And it's something we don't talk a lot about here at LifeVantage, but really what is the most important thing to get that sirtuin activity is actually the ratio of NAD to NADH. And that ratio is important because it is the same molecule, but one is oxidized and one is reduced. Okay, so we want the oxidized NAD plus to actually increase. And so this data is showing exactly that. So we saw the increase in NAD, a slight decrease in NADH, although still significant. And again, because these error bars are so tight. And then also the ratio improved. So in every aspect, in these cells in culture, this theacrine-based supplement increased NAD levels. And here they just took a picture of the plate showing that they could measure NAD plus and NADH. Then we jump into the discussion, and here, like I said, they are going to be putting all of this research into the larger context of the scientific literature, but they also talk about the limitations of the study. So it's always funny to read these because you have to, in your literature, in your paper, talk about what are some of the limiting factors to your study. So I've underlined here on my hard copy. I'm a little old school. I still like to print them out and read it. But here they talk about some of the shortfalls. Oh, why aren't you highlighting for me? Well, in here. So they talk about, let me highlight now, but NAD3, right, this core complex was able to increase global sirtuin activity, and the gene expression in part like agrees with the scientific literature. For instance, then they give examples of why they feel it's supported. But then they start talking about limitations. So due to cell lysate constraints, we opted, right? So then they start to talk about what some of the flaws of the study might be. But then there's always a but statement in all of these, which I always laugh at. But then they say, however, but this is why our research is still valid. And you'll see that theme in every paragraph in the discussion. They got a little more into kind of the limitations of their work. But again, you'll still see there's that but statement again, notwithstanding, but these preliminary findings regarding the effects of the theacrine-based supplement in global sirtuin activity, this rate-limiting enzyme, NAMPT, and then most importantly, those cellular NAD levels. So in my mind, with this work that came out now a couple years ago, perfectly fine to say Protandim NAD synergizer is been shown to increase NAD levels inside of the cell. And then the conclusion, those are usually always really straightforward. These can be interesting reads down here. Nowadays, you have to disclose any conflicts of interest. So I'd say this research study was conducted on behalf of LifeVantage Corporation. Brian Dixon is an employee of LifeVantage Corporation. So always good just to make sure there's nothing too fishy going on with their conflicts of interest. And then we're down to the references. And again, those are only there in case you want to double check their work or maybe go learn some more background or on another topic yourself. So that's kind of my take on how I approach scientific works, those different sections. And then I think this really cool study that supports the efficacy of one of our amazing Protandim family of products.
Actually, two quick questions, Dr. Dixon. First, in this study, I didn't see the name Protandim, NAD Synergizer, or LifeVantage anywhere. Is that deliberate so that there is no conflict of interest, or what's the reasoning behind that?
Yeah, that is deliberate. And it will also be interesting when the patents are fully accepted with the US Patent Office. You know, the language we use in there is much different than patents of the past because we want to make them more compliant. So, you know, we're not talking a lot about disease. We're not talking a lot about some other things that have gotten us in trouble in the past. So moving forward, that's one thing we're working closely on. You know, this work was done with a strategic partner of ours, and we've done our own research at our own laboratories, and we're able to like almost exactly to a T repeat their work. So in two different laboratories, they're literally different sides of the country, we were able to corroborate their work. But yeah, Dundee, we definitely leave that stuff off on purpose. It's deliberate.
Okay. Next question, and I'm not sure how you can answer this, but or what relevance it has to our quote unquote competitors. But with regard to Dr. David Sinclair, who obviously, I mean, for people who know about NAD, he's a very big proponent on it. He talks about it all the time, and he has intellectual property rights to two products that we would consider our competitors. If when someone asks us, because I get asked this question pretty commonly, how does the Protandim NAD product compare to those other two products? And would it be okay to mention them? I don't want to do any direct correlation or get us in any trouble, but I do get asked about them often.
I think the best way to describe all of those nutrients that you were referring to would be precursors. And so in a biochemical pathway, I mean, if you think about it, it really is a factory assembly line. It's taking raw materials, whether they come from the diet or our metabolism, and it's making whatever molecules the cell needs for its day-to-day functioning. So I love the factory analogy. So if you think about an assembly line, right? An assembly line can only work as fast as the slowest worker. So the same thing is true in these biochemical pathways. This biochemistry is only happening as fast as the slowest enzyme will allow. So when I talk about these raw material aspects, and there's 3, 4, 5 of them out there on the market, that approach works to a point, but at some point you can pile up all the raw materials you want outside the factory, but you're only able to utilize them as quickly as that assembly line will work. So at some point, you've just overwhelmed the system and you're not going to get any additional benefit from it. Our approach using nutrigenomics is to not try to force these chemical reactions to go, but rather let's build additional assembly lines inside of the cell, right? Because if I had one, and if I'm able to put another one inside of it, I've literally doubled the output of that cellular factory. And so that's our approach. Let's make more assembly lines or more of these factories inside of the cell to make more NAD, instead of just giving people these mega doses of these precursors. And of course, the best approach would be to utilize both. You want to make sure you have enough precursor around. In this case, it's just a B vitamin, nicotinamide, that's easy enough to get from the diet, but we've also put a little bit of niacin in there as well to help support that function.
Great explanation. Thank you so much. It just, to hear it put in that really simplistic example of an assembly line, just makes me personally more proud of our product because of how superior it is actually when you look at the comparison. Thank you so much for that.
Oh, my pleasure. My pleasure. And what about any other questions you can think of? Something I might have missed? And sorry, Don D, I will have a hard stop here at 1, but
I'm looking through the chat and seeing. Marisa Maggio Harrelson asked, who conducted the research and how did it go about?
Yeah, absolutely. So that's where we can get that information right from the article. So if I share that article again, it will be up. It'll be in two places. It'll be up in the author list. So you'll see the 1, 2, 1, 1. These are their affiliations. So here, the School of Health Sciences, Linwood University out in Missouri. And then also at Auburn University. And then if we go back to that kind of conflict of interest statements, we can find out more there as well. So where did the funding come from? What were those requirements I was talking about, that each author has to actually do something unique in the study. So this tells us what specifically the authors did. Yeah, like I said, where the funding came from, and then their conflicts of interest. So really easy to find in any research study.
That was kind of interesting at the end of the discussion when you were talking about the but statement, or actually the first line of the conclusion, when it says, however, and as mentioned prior, human studies are needed to validate the current findings. So I love it when that's actually a positive. Is obviously the results here were beneficial enough to now go from a mouse model to a human model, but the results are compelling enough to make it worthwhile. So that's awesome. I love to see that. And that's one of the things that people don't quite understand sometimes is when you're putting together a study, well, why aren't there any human studies? Well, there's a hierarchy. It goes from, animal cells and then to tissue cells. And if it's compelling enough of an argument or a discovery, into human trials, but it's usually along that process. So just because we don't have a lot of human trials yet, doesn't mean we have products that are inferior to anything else out there, because there's that natural progression. It starts with animals, then goes to cells, and then to humans. So love that.
Absolutely. And yeah, you'll do a lot of quick and dirty research in, let's say, cell culture. And then, yeah, you hope to move it in. But they also did a beautiful job there of setting themselves up, because they knew this human clinical study was already underway. And now we know the papers are coming out. So they've already started answering their own questions.
Yeah. Can you take another question from the chat?
I do. I've got to run on to another webinar though, Dr. Nettles. I apologize.
Okay. No
I'll do one more quick one.
Okay. And it's from Team Knox. Says, how does our product compare to intravenous NAD, peptides, et cetera, that relate to elite athletes?
I have a pretty negative bias towards IV NAD injections. From trying to actually measure NAD levels in my experiments. NAD is incredibly unstable. It's actually incredibly difficult to measure, because remember, half of it's oxidized and the other half's reduced. So that means it's very reactive. And so depending on how you're handling it, it's going to either oxidize or reduce. I have a hard time imagining a scenario where any of that NAD is really making it to where it needs to be to actually get into tissues and do its job before it gets destroyed. But I'm open-minded and I can be convinced otherwise, but that's my initial take on it.
Sure sounds like flipping the NAD switch in your body and letting your body do the rest sounds like a better way of doing it. Thanks so much for that, Dr. Brian.
Absolutely.
Well, thank you everybody for getting on the call today. Points on product. I know Dr. Dixon's got to go, but thank you, Patty. Thank you, Greg, who's not here, but thank you guys for the trust and confidence in me to be able to host this call. And Dr. Brian, thank you so much for your